Welcome to the deep dive. Today we're jumping straight into something. Well, something that really challenges a lot of common assumptions in neuroscience. It's about the brain, specifically how male and female brains might be organized.
And for a long time, the story most people heard, and frankly what a lot of science suggested, was this idea of a mosaic brain.
Right, the mosaic idea, meaning that while there might be average differences, any individual brain is a mix of features, and you couldn't reliably look at one scan and say, that's male, or that's female. Overlapping features, high variability.
Exactly. That's been the dominant narrative. Lots of overlap, very hard to distinguish individuals.
Okay, but here's the kicker, the paradox we really need to unpack today. If that mosaic idea is the whole story, how on earth can a new deep learning model, fed only functional brain scans, predict biological sex width? Get this. Over 90% accuracy.
That's the question, isn't it? It's a massive contradiction. 90% accuracy is, well, it's staggering if you believe the brains are fundamentally indistinguishable at that individual level.
So what gives? Are the differences actually minimal, like the mosaic theory suggests? Or have our older scientific tools just maybe not been sharp enough to see the more subtle dynamic patterns?
That's what this deep dive is all about. We're looking at a study that used some really clever tech and, crucially, massive amounts of data to argue that maybe our tools were too blunt.
And our mission today isn't just to report that 90% number. We want to get into the weeds, understand the technology, this novel neural network they built, and figure out what differences it's picking up on, where they are in the brain, and the big one, why this level of accuracy seemed impossible for so long.
Yeah, and the absolute key, as you hinted, is dynamics, moving beyond just static pictures.
Explain that, static versus dynamic.
Well, think of older methods looking at brain connectivity. Often they'd average the connections over, say, a whole 10-minute scan. You get one overall picture, a static map.
Like a single photograph of traffic flow averaged over an hour.
Kind of, yeah. But this new study focused on the dynamic functional organization, how brain connections fluctuate and change from moment to moment. The rhythm, the flow, the conversation between brain regions as it happens.
So not the average traffic, but the real-time ebb and flow.
Precisely. And that shift from static averages to dynamic patterns seems to be the technical leap that unlocked these really striking findings.
Okay, let's get into the study itself. To really feel the weight of that 90% number, you need to grasp the scale of the data they used. This wasn't just a handful of scans.
Oh, definitely not. When you're tackling something this potentially controversial, you need numbers, big numbers. And you need to show it's not just a one-off. So they pulled data from 3 huge independent groups of young adults, mostly in the 20 to 35 age range.
And the main training ground for their model was the Human Connectome Project, the HCP.
That's right. The HCP is sort of the gold standard for high-quality brain imaging data. Just for one session they analyzed, they had resting state fMRI from 583 females and 490 males. That's already a pretty big sample for neuroimaging.
But the HCP, great as it is still just one project, one set of scanners, one main demographic profile, more or less.
Exactly. And that's why the other data sets are so critical for validation. They didn't just stop with HCP. They tested their model on 2 completely separate cohorts.
Which were?
The Nathan Klein Institute Rockland Sample, NKIRS, and the Max Planck Institute Leipzig Mind-Brain-Body dataset. These came from different places, used different scanners, different protocols, different mix of people.
So if the model trained only on HCP data still works well on these completely different data sets,
then it strongly suggests the patterns it found are real fundamental differences in brain organization, not just some weird artifact of the HCP scanner or the specific people in that study. It points towards generalization.
Okay, let's talk about the tech itself. The SDDNN, that's the star player here, right?
Yeah.
Spatiotemporal deep neural network. What makes it special? Why do standard deep learning methods often struggle with fMRI?
Well, the core problem is the nature of fMRI data itself. It's inherently 4-dimensional. You've got the 3 dimensions of space, the brain volume, plus the 4th dimension, time. Brain activity is constantly changing.
Right. And deep learning models, especially things like 3D convolutional neural networks, they're great with spatial data, like images. But 4D.
It just explodes computationally. The number of parameters you need to train becomes enormous. And here's the other constraint. Neuroimaging datasets, even big ones like HCCP, are tiny compared to the millions or even billions of images used to train models for, say, self-driving cars or image recognition.
So you have this super complex data structure, 4D, colliding with relatively few examples to learn from. That sounds like a recipe for failure for standard deep learning.
It often is. You either can't train the model or it massively overfits the training data and doesn't generalize. The SDNN's innovation is how it handles this. It essentially reconceptualizes the problem. Instead of trying to process the whole 4D volume directly, it treats the fMRI data more like a multivariate time series. Think of it as tracking the activity signals from many different brain regions over time. And crucially, it uses 1D convolutions.
One-dimensional. So the convolution filters, the things that detect patterns, are primarily sliding along the time axis.
Exactly. They slide along the time dimension for each brain region's signal. This makes it much, much more efficient at capturing the temporal correlations, how the activity in different regions waxes and wanes together, how their functional connectivity changes dynamically over time.
Oh, okay. So it's optimized for the time dimension, which is where the dynamic action is happening. It's still capturing spatial info implicitly because it knows which region's time courses it's comparing, but the computational heavy lifting is on the temporal patterns.
Precisely. It models that dynamic connectivity, the time-varying conversation between regions, which, as we'll see, is exactly what the older static methods were averaging out and missing.
That shift is clever. But even then, you said the data sets are relatively small for deep learning. 1,000 subjects from HCP still sounds challenging. How do they boost the data?
Right, that's the second piece of the puzzle, data augmentation. Even the efficient SDDNN needed more examples to learn robustly. So they used a technique called a sliding window approach.
Sliding window, like chopping up the data.
Sort of. Imagine the full fMRI scan for one person is like a 15-minute movie. Instead of just feeding the whole movie into the model once, they took shorter segments, say a 5-minute clip.
Okay.
Then they slid that 5-minute window forward in time, maybe by just 1 minute, and took the next overlapping 5-minute clip. And repeated this across the whole scan.
Ah, I see. So from one 15-minute scan, you generate lots of slightly different 5-minute examples. Each example still has the same label, male or female, but the model sees many more variations of the dynamic patterns within that person.
Exactly. It dramatically increases the effective number of training samples. The paper mentions this boosted their training data set size by almost 15 times, going from around 800 subjects per session in the training set to something like the equivalent of 12,000 data segments to learn from.
Wow. Okay, that's a serious power-up. Necessary to train such a deep model without it just memorizing the initial data.
Absolutely essential for generalization.
So you combine this clever SDDNN architecture focused on temporal dynamics with this massive data augmentation.
Yeah.
And you apply it to high-quality HCP data. What was the result?
The headline finding. Using a standard brain map, the BrainTome Atlas, the SDDNN model, achieved 90.21% accuracy in predicting sex from the resting state fMRI data. And the AUC, another performance metric, was 0.97, which is incredibly high.
90%, just from watching the brain's resting activity patterns over time.
And that was just the baseline. They did robustness checks using different ways to define brain regions, other atlases like Shen 268 or CC400, which carved the brain up differently.
And did the accuracy hold up?
It actually got slightly higher. They reported accuracy up to 92.17% and 91.98% with those other atlases.
Okay, that's important. It suggests the finding isn't just an artifact of one specific way of mapping the brain. The dynamic difference seems to be a real feature, regardless of the exact map you use.
Yeah, it shows robustness. The signal is strong enough to be picked up consistently.
All right, so you get a result like 90% plus accuracy. Immediately, anyone in the field is going to raise an eyebrow, right? The skepticism kicks in.
Oh, absolutely, and rightfully so. The first questions are always, is this real? Is it just specific to your fancy HCP data set, your specific scanner, your particular group of people? Can anyone else find this?
Which brings us squarely to section 2, generalizability. Showing high accuracy on the data you trained on is one thing. Showing it works on completely new, unseen data from different places. That's the real test of a scientific finding.
Precisely, and this is where those other two cohorts, NKIRS and MPI Leipzig, become absolutely critical. Remember, the model was trained only on HCP data.
Right, no peeking at the other data sets during training.
None at all. They just took the trained HCP model and applied it directly to the brain scans from NKI and MPI. Different people, different scanners, different research groups.
What happened? Did the accuracy plummet?
It dropped a bit, as you'd expect when moving to totally new data, but it was still remarkably high. The generalization accuracy consistently landed somewhere between 82% and 86% across those independent cohorts.
Okay, wait, so even on completely different data sets it hadn't seen, The model could still tell male from female from brain activity dynamics over 80% of the time.
Yes. And while 82% isn't 90%, scientifically, maintaining that level of accuracy on independent validation cohorts is, well, it's huge. It really argues against this being just some quirk of the HCP data.
It suggests that dynamic patterns the SDDNN learned are genuinely generalizable features of how young adult male and female brains function differently, at least in terms of resting state dynamics.
That's the strong implication, yes. It's not just noise or a dataset-specific artifact.
This leads us to the next layer of skepticism, the anti-junk controls, as you called them earlier. In brain imaging, especially fMRI, there's always the worry about noise, artifacts, people moving slightly in the scanner. Could the model just be picking up on that junk?
It's the classic critique, especially for connectivity studies.
Yes.
So the researchers were really thorough here. They systematically tested robustness against the usual suspects.
Like what?
Well, we already mentioned they checked across multiple brain atlases, 8 different ones actually, like AAL, Glasser, Shen. If the effect vanished when they changed the map, that would be a red flag. But it didn't. Accuracy stayed high. Check 1.
Okay, what else?
They also tested different ways of cleaning the data. There are various statistical methods neuroimagers use to remove noise signals, like those caused by breathing, heartbeats, or tiny head movements, things called a comp core or motion scrubbing.
And the model's performance
Remain high regardless of which cleaning pipeline they used. So it wasn't dependent on one specific way of pre-processing the data. Check 2.
makes sense. But the big one, the potential killer, confound in resting state fMRI, is always head motion, right? Even tiny systematic differences in how much males versus females move could create fake connectivity patterns.
Absolutely.
Yeah.
If, say, males in the sample consistently move their heads just a tiny bit more than females, a sensitive deep learning model might latch onto that movement signal thinking it's a biological sex difference when it's really just motion.
This has sunk studies before, hasn't it?
Many times. So they tackled this head-on in 2 main ways.
Okay, first.
First, the simple check. Did males and females in these specific cohorts actually move differently on average? They calculated the standard measure, mean frame-wise displacement. No significant difference in any of the 3 cohorts, HCP, NKIRS, MPI Leipzig. There wasn't a statistically significant difference in average head motion between males and females, so a systematic motion bias was unlikely to begin with.
Okay, that's good baseline evidence. But maybe the pattern of movement matters, even if the average amount is the same. Or maybe the specific features the model is using are somehow subtly linked to motion artifacts.
Exactly. They couldn't just stop at the average. They needed to check if the information the STDNN was using for classification was independent of motion.
How do you even check that?
They used a pretty sophisticated statistical tool called square distance correlation, or DCOR2. It's designed to measure the statistical independence between 2 potentially complex sets of variables.
So in this case, the variables were?
On one side, the specific dynamic brain features the STDNN identified as important for predicting sex. On the other side, various metrics quantifying head movement. They calculated the DCOR2 between these 2 sets.
And if the features the model used were independent of motion, the DCOR2 value should be?
Close to 0. A value near 0 indicates statistical independence. The brain features the model relies on are not carrying significant information about head movement.
And the result?
The DCOR2 values were indeed near 0. Consistently across all sessions and cohorts.
Wow. Okay, let's translate that. It means the stuff the model was using to get that 90% plus accuracy was not significantly related to how much people were moving their heads in the scanner.
Precisely. It effectively rules out head motion as the primary driver of the classification accuracy. This is a really critical control. It strongly suggests the accuracy comes from genuine differences in functional brain organization, not just movement artifacts.
That's a very thorough piece of validation. It really shores up the core finding.
It does. It addresses one of the most persistent and valid criticisms leveled at fMRI connectivity studies.
Okay, so we've got high accuracy, it generalizes to new data sets, and it's robust to artifacts and head motion. The difference seems real. But now, the black box problem. The SDNN model knows how to classify, but what is it actually looking at? What are these dynamic features?
Right. Getting beyond just the accuracy score and understanding the why is the next crucial step. The researchers needed a way to interpret the STDNN, to peek inside and see which specific dynamic patterns were most influential.
How do they do that?
They used a technique called integrated gradients, or IG, It's a feature attribution method. Essentially, it allows you to trace the prediction back through the network and highlight which input features in this case, which patterns of dynamic connectivity between specific brain regions over time were most responsible for pushing the model's decision towards male or female.
So IG acts like a spotlight, illuminating the key dynamic interactions the model learned were important. These are the brain fingerprints you mentioned earlier.
Exactly. These highlighted dynamic patterns are the specific features that constitute that individual's functional fingerprint, the ones driving the classification.
And speaking of fingerprints, you mentioned something earlier about individual stability that sounded pretty amazing.
Yeah, this was a fascinating side analysis they did. They looked at how stable these individual dynamic fingerprints were within the same person over time. They had data from 2 different scanning sessions for many individuals in the HCP dataset.
So they could compare session 1's fingerprint to session 2's fingerprint for the same person.
Precisely. And they asked, is my dynamic brain signature from session 1 more similar to my own signature from session 2, or is it more similar to someone else's signature from session 1?
And the result?
It was stunningly consistent. For over 99.5% of individuals, their own dynamic features from session 1 were statistically more similar to their own features from session 2 than they were to the features of any other person in the dataset.
Wait, 99.5%?
Yeah.
That means your brain's dynamic functional rhythm is incredibly unique and stable over time, like a personal signature.
It really suggests that. The specific way your brain regions dynamically communicate, that time-varying dance, is highly individualized and consistent, at least over the timescale of these sessions. The STDNN isn't just picking up random noise, it's identifying a stable, deeply personal characteristic of brain function.
That stability really adds weight to the idea that these features are biologically meaningful. Okay, so the fingerprints are stable and individual. Now the big question, which parts of the brain, which regions, are consistently involved in creating these sex-differentiating fingerprints? Did they find a consensus across the different data sets?
Yes, they synthesized the findings from the integrated gradients analysis across all 3 cohorts, HCP, NKIRS, and MPI Leipzig, to find the brain regions whose dynamic connectivity patterns consistently contributed most to the sex classification.
So what are the key players?
They found that the contributing regions clustered mainly into 2 broad functional networks. First, there were significant contributions from prefrontal components.
Prefrontal cortex, that's like the brain's control center, right?
Very much so. We're talking about areas like the dorsolateral prefrontal cortex, both right and left sides, the DLPFC. Think of this as the seat of executive function. Working memory, planning, complex problem-solving, cognitive control.
Okay, DLPFC. What else in the prefrontal area?
Also involved were parts of the ventrolateral prefrontal cortex, the VLPFC, and the ventromedial prefrontal cortex, the VMPFC. These areas are more heavily involved in things like emotional regulation, decision-making based on value, and processing social and emotional cues.
Wow. So the differences aren't just in some obscure sensory area. They're showing up consistently in these high-level control and regulation hubs. DLPFC, VLPFC, VMPFC.
Exactly. Regions critical for how we manage information, make choices, and handle our social and emotional worlds. That's a pretty profound finding in itself.
Okay, that was the first cluster prefrontal components. What was the second main group?
The second group involved key posterior and subcortical components, primarily regions like the posterior cingulate cortex, or PCC, and the precuneus.
Ah, the PCC and precuneus. Those are the major hubs of the default mode network, aren't they?
That's right. The DMN is most active when our minds are turned inward, thinking about ourselves, remembering the past, planning the future, mind-wandering. It's about internal, self-referential thought.
So we have dynamic differences linked to external control and regulation via the prefrontal cortex, and differences linked to internal thought processes via the default mode network hubs.
It seems so. They also noted contributions from parts of the temporal lobes, like the superior and middle temporal gyri, which are involved in processing sensory information and language, and even subcortical structures like the striatum, specifically the dorsal caudate nucleus, involved in habit learning and motor control, and the thalamus, which is like a central relay station for almost all sensory and motor information going to the cortex.
Okay, so it's not just 1 or 2 spots. It's a distributed network involving dynamic coordination between executive systems, internal thought systems, and even sensory motor relay points.
Yes, it appears to be a difference in the large-scale dynamic functional architecture, not just localized activity.
All right, finding a real, robust, replicable difference in brain function like this is already a huge deal. 90% accuracy, stable fingerprints, key regions involved. But the ultimate question is, does it matter? Does this difference in dynamic brain organization actually relate to anything tangible, like how people think or behave?
That's the million-dollar question, isn't it? Moving from brain scans to real-world relevance. And yes, the study tackled this directly. They investigated whether these dynamic sex-discriminating features identified by the SDDNN could predict individual differences in cognitive abilities and behavioral traits.
How did they measure cognition and behavior?
They used the rich behavioral data collected as part of the Human Connectome project. It includes scores from a whole battery of tests, 14 different measures in total.
14? Like,
what? All sorts of things. Standard cognitive tests measuring, say, episodic memory, fluid intelligence, processing speed, but also measures related to personality and self-control, like assessments of self-regulation and impulsivity.
okay, 14 different scores. That's a lot to correlate with complex brain patterns. How do they simplify that?
Good point. Analyzing all 14 separately could get messy. So they used a statistical technique called principal component analysis, PCA, to boil those 14 measures down to their essential underlying factors.
And what did the PCA find? What were the core components?
It extracted 3 main principal components, or PCs, that captured most of the variance in the behavioral data. The first PC was strongly linked to measures of general intelligence IQ, basically, covering fluid intelligence and vocabulary.
Okay, PC1 is like general cognitive ability.
Right. The second PC seemed to represent behavioral inhibition, things like cognitive control, the ability to suppress inappropriate responses. And the third PC was related to reward-related self-regulation, which taps into impulse control and decision-making, especially when potential rewards are involved.
So IQ, inhibition, and reward regulation. Those are the 3 core behavioral profiles they looked at.
Exactly. Now, the crucial test. Could the dynamic brain features predict where individuals landed on these 3 cognitive profiles? And more importantly, was the prediction the same for males and females?
How did they test that link between the brain features and the cognitive profiles?
They used another sophisticated technique called canonical correlation analysis, or CCA. You can think of CCA as finding the strongest possible mathematical relationship, the optimal correlation between one set of variables, the dynamic brain features from the SDDNN, and another set, those 3 cognitive profile scores.
Okay, so they ran this CCA using the dynamic features from the SDDNN model.
Yes, the high-resolution, time-varying fingerprints. And crucially, they performed the analysis separately for the male participants and the female participants.
And the result? This feels like the climax.
It really is the core payoff of the whole study. The CCA revealed sex-specific neurobiological predictors of the individual cognitive profiles.
Whoa. Okay, unpack sex-specific neurobiological predictors. What does that mean in practice?
It means the way the dynamic brain features mapped onto the cognitive scores was different for males compared to females. The functional architecture underlying those cognitive profiles seemed to be organized differently.
So, for example, the pattern of dynamic connectivity in, say, the DLPFC that best predicted IQ score in females was not the same pattern that best predicted IQ score in males.
Essentially, yes. While some of the same regions were important predictors in both sexes areas, like the PCC and DLPFC are fundamental for complex thought, after all the specific multivariate relationship, the way the dynamic signals from multiple regions combined to predict the cognitive score was unique to each sex.
The functional recipe was different.
That's a good way to put it. The specific weighting and timing of the ingredients, the dynamic connections that produced a certain cognitive outcome, seemed to be organized differently. Maybe the link between VMPFC dynamics and reward regulation works one way in males and is weighted differently in females. The STDNN features captured this sex-specific functional organization.
Okay, that's a huge finding. The dynamic features reveal sex-specific brain cognition links. But the researchers didn't stop there, did they? They ran a critical control analysis using the old way of looking at connectivity.
Exactly. This is where the contrast becomes crystal clear. They repeated the entire brain cognition prediction analysis, the CCA, but this time, instead of using the dynamic features from the SDNN, they used traditional static functional connectivity measures.
The old method connectivity averaged over the whole scan, the flat snapshot.
Right. The standard approach used in countless previous studies. And what happened when they used static connectivity to predict cognitive profiles?
Let me guess. The sex-specific finding disappeared.
Completely. The static functional connectivity models failed to uncover any significant sex-specific neurobiological predictors.
Wow. So when you average out all those dynamic fluctuations, you lose the sex-specific signature linking brain to behavior.
That's what the data shows. Instead, the static connectivity models only identified brain features that were sex invariant,
Right.
meaning the static brain features that predicted cognitive profiles in males also predicted cognitive profiles in females, and vice versa. The relationship was the same for both sexes. The prediction model was transferable.
Okay, this is really the crux of it, isn't it? Static connectivity reveals the shared foundation, the parts of the brain important for cognition in general, across both sexes.
Exactly, like the basic blueprint of the house, which is largely the same.
But the dynamic connectivity, captured by the STDNN, reveals the specialized wiring, the specific flow of information, the real-time operations. And that dynamic organization appears to be specialized differently by sex, particularly when linking to complex cognitive functions.
That's the major implication. The dynamic organization seems to hold the key to understanding these sex-specific brain behavior relationships, while static organization masks them.
This really reframes things.
Yeah.
It suggests that maybe years of research relying only on static measures might have systematically overlooked some really important functional specialization simply because the tools weren't sensitive to these fast, time-varying dynamics.
It certainly raises that possibility. The most behaviorally relevant differences might lie precisely in that dynamic functional rhythm.
Okay, so we have this powerful finding. 90% plus accuracy, replicable, robust, individually stable fingerprints, key brain regions identified, and critically, these dynamic features reveal sex-specific links to cognition that static measures miss. It's compelling.
No.
But we have to talk about the caveats, the limitations, right? Science demands nuance.
Absolutely essential, especially with a finding this potentially impactful and, let's be honest, potentially controversial. The researchers themselves are very clear about the limitations in their discussion. We need to be just as clear.
What's the first major caveat?
Potential bias in the data. Even though they replicated across 3 diverse cohorts, HCP, NKI, MPI, which is a huge strength, there's always a possibility that hidden biases related to things like ethnicity or maybe socioeconomic status, which can correlate with biological sex and sample populations, might be subtly influencing the functional patterns the model picks up on.
So the model might be detecting something real, but it could be partially reflecting cumulative environmental or social factors linked to those demographics, not just biology.
It's a possibility that needs to be kept in mind, even with the strong generalization results. Science needs to remain cautious about interpreting the source of the difference.
Okay, what's another key limitation?
This is a really important one for everyone listening to grasp. The model predicts biological sex based on brain scans from young adults, period. It does not account for the full picture of sex and gender. Specifically, the study didn't include measures of hormonal influences. We know hormones like estrogen and testosterone fluctuate and can significantly impact brain function and connectivity, potentially contributing to these dynamic patterns. That wasn't measured here.
And critically, it didn't measure gender identity either, did it?
No. Gender identity, one's internal sense of being male, female, both, or neither, is distinct from biological sex assigned at birth. This study is about the biological sex classification based on brain function patterns observed in large groups assigned male or female at birth. It cannot and does not say anything about the neural correlates of gender identity itself.
So the 90% accuracy is about a statistical difference in brain function between biological sexes in this age group. It's not some kind of rigid, deterministic male brain versus female brain stamp that dictates psychology or identity.
Absolutely not. It's a probabilistic difference in dynamic functional organization. High accuracy reflects a strong statistical pattern across groups, not an inevitable destiny for any individual. Misinterpreting this as rigid or deterministic is where the real danger lies.
Which leads naturally into the future directions the authors call for. They've shown that these dynamic, sex-specific differences exist and link to cognition, but not how they arise.
Exactly. The paper explicitly calls for more research to understand the origins and precise roles of these sex-specific functional patterns. They highlight the need to investigate factors like, well, genetics, obviously, but also developmental trajectories. How do these patterns emerge over childhood and adolescence?
And importantly, they also mention learning strategies and psychosocial influences.
Yes, this is critical. How much are these dynamic patterns shaped by experience, by the different ways society might encourage or reinforce certain cognitive strategies or emotional regulation styles in boys versus girls, men versus women?
Because the brain is incredibly plastic.
Right.
It adapts to the demands placed on it. If social roles or cultural expectations consistently place different demands on, say, executive function or emotional processing systems, depending on sex.
Then it's entirely plausible that the brain's dynamic functional architecture would optimize and specialize differently over a lifetime to meet those demands. That lifelong shaping could absolutely contribute to the sex-specific patterns the SDDNN is detecting.
So the observed 90% plus accuracy could reflect a complex interplay of innate biological predispositions and decades of socially and culturally sculpted learning and adaptation.
That's likely the most realistic picture. The brain is not static. It's constantly shaped by both biology and experience. Teasing apart those contributions is the massive challenge for future research. The difference the SDNN found is real, it's stable, it's behaviorally relevant, but its ultimate origin story is still being written.
Okay, let's try and weave this all together then. What's the core takeaway from this deep dive?
I think the main point is that this novel deep learning approach, the SDDNN, by focusing specifically on dynamic, time-varying brain connectivity, has uncovered robust, replicable sex differences in functional brain organization that reach over 90% classification accuracy.
And these differences aren't trivial. They're found in key brain networks controlling executive function, internal thought, and emotional regulation. And crucially, they seem to be linked to individual cognitive profiles in a sex-specific way.
Right, a link that was completely invisible when using older static measures of connectivity. Static methods only picked up the shared sex-invariant features, suggesting the real specialization lies in the dynamics.
We also learned that these dynamic patterns are incredibly stable within an individual over time, like a personal functional fingerprint, identified with over 99.5% consistency.
Yeah, that individual stability is remarkable.
Which brings us inevitably to the final provocative question we always like to leave you, the listener, with. Given that we now have strong evidence for these stable, individually unique, and behaviorally relevant sex differences in dynamic functional brain organization.
The big question isn't really if these differences exist anymore. The SDDNN study makes a compelling case that they do, at least in terms of functional dynamics. The deeper, more complex question now is, how much of this specialized dynamic organization is laid down early by genetics and hormones? And how much is actively sculpted over a lifetime by the profoundly different social roles, cultural expectations, and learning experiences that males and females often encounter?
That's the edge where the neuroscience currently stands, and where the much broader societal discussion needs to continue. Nature versus nurture, biology versus experience. The answer for the dynamic brain is likely, as always, a complex conversation between the two, something for you to mull over.